Okay, so we're going through biological molecules. We talked about carbohydrates. Next up is lipids, fats. And the defining characteristic of fats is that they are what's called non-polar. Remember when we talked about polar covalent bonds? We talked about they had a positive charge and a negative charge. Water has these polar covalent bonds, so water is polar, and therefore it has has that region where it's positively charged and a region where it's negatively charged. The adjacent molecules attached to each other. It makes it a good solvent of polar molecules, of ions, right? Sodium, salt gets broken up because salt is ionically bonded. It's got a positive and a negative. The water molecules can get in between them. Salt dissolves. Carbohydrates have polar regions, have positive, negative regions, so they are polar. They can be dissolved in water. Lipids are not. So they don't dissolve in water. That is they are hydrophobic. Fats are hydrophobic. They won't dissolve in water. They want to get away basically from water. They don't mix well with water, and you've seen that oil and water don't mix. Now, for anatomy and physiology, for biology, what we're talking about, what are we talking about when we talk about fats? Where some examples of fats are something called fatty acids, something called neutral fats. Those are going to be the things that we're talking about when we talk about biological systems. There's oils and steroids also. We're going to be talking about those a little bit in biological systems. Steroids are important. So let's start with talking about fatty acids. So these are a type of lipid, a type of fat. They're called fatty acids. And basically what fatty acids are, they're organic molecules, so they're going to be carbon and hydrogen. And these are basically long chains of carbon. Carbon can form 4 bonds. So you've got a carbon, it's got 4 bonds. It's bonded to a carbon over here. It's bonded to a carbon over here. It's bonded to a hydrogen over here. It's bonded to a hydrogen down there. And it's just a chain that just goes on and on and on and on. And at the end, it will have with a carboxylic acid end, CO and an OH kind of thing at the end. Those are fatty acids, these huge chains of carbon and hydrogen. And we're going to be seeing these fatty acids in a moment. They're going to be important. This would be, now, when we talked about carbohydrates and when we talk about proteins and when we talk about nucleic acids, we talk about monomers and polymers. Lipids don't really form polymers because fatty acids are sort of, I guess, would be of biological lipids. They would be the monomer, the small part. But you'll see that you can make long fatty acid chains, but you can only make a lipid molecule with maximum 3 fatty acid chains. We'll talk about that in a second. Now, fatty acids, our bodies make fatty acid chains. We make them. And they have isomers, cis isomer, trans fats. You may have heard of trans fats. Whether they're good for you or bad for you is still under debate. A lot of food science is not very good. It's sponsored by companies and stuff like that. Some of it is really good, but a lot of it is not good, mainly because it's hard to do controlled experiments on humans. How do you deny humans food? How do you make a controlled experiment? Fatty acids, big long chains, carbon, hydrogen. They can have an isomer, cis isomer, trans isomer, and they can be saturated, you may have heard this, or unsaturated. Unsaturated fats and saturated fats. On your chip bag, it might say no trans fats or saturated fats or something like that. Basically, what saturated means, so when I describe to you the carbon with a hydrogen down there and a hydrogen down there, that carbon is saturated. All 4 bonds are attached to an atom. Hydrogen down here, carbon down here. But they can be unsaturated, which means that instead of hydrogen being up here, for example, there's a double bond with the carbon next door. And the carbon next door, therefore, is missing a hydrogen. You can make a double bond. And remember, these are covalent bonds, covalently bonded. And so the more of these double bonds you have and the less hydrogens you have, the more unsaturated the fatty acid. There's an example here. Let's look at it. Zoom in there. We're gonna zoom in there. We'll see it. Yeah, there we go. Perfect. So this is a saturated fatty acid. See, there's that carboxylic acid and COOH. And then all of these carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. The last one has carbon and 3 hydrogens. An unsaturated fatty acid has a double bond here. And so the adjacent 2 carbons don't have that hydrogen. You can see a kink forms in the molecule. And remember, shape is important. We're going to talk about more about it, but shape is important. So here we've got, the shape is slightly different. And you can have lots of unsaturated, because you can have a lot of those carbons are unsaturated. Now, when we make fatty acids, when we make fatty acids, what ends up happening is we make saturated fatty acids. And one of the properties of saturated fatty acids is that they are solid at room temperature. Okay, so I guess butter would be solid at room temperature, whereas margarine isn't. So margarine is more unsaturated than butter. Unsaturated fatty acids, which are considered the bad ones. I don't know why they're considered bad ones, but think of like vegetable oil. These are made by plants. So plants make more unsaturated fats. I think this whole saturated, unsaturated thing, as far as I know right now, with the current science, is it doesn't matter. You can eat unsaturated fatty acids. I think you're okay. And those are liquid at room temperature. Again, vegetable oil, margarine is a little bit more liquidy. So those are the things to remember about fatty acids, okay? And be able to recognize a fatty acid. Sure, you see a long chain of carbon hydrogen with a COOH at the back. That's a fatty acid. Next so then what we can do with these fatty acids is we stitch them together. So those are like sort of the basic building block. And what we do is we stitch them together, but we don't stitch the fatty acid to the fatty acid. That would just make like a longer fatty acid. What we do is we cleave off we cleave off part of it, and we attach it to an alcohol called a glycerol. I know, but just bear with me. So let's look at what a glycerol looks like, if you can see it. Let's move away here. So glycerol is 3 carbons. It looks kind of like a fatty acid, except it's got all these OHs. And when you see these OHs, remember, that's where we can do dehydration synthesis, right? We can do dehydration synthesis. And what we can do, we could take a fatty acid. So this is a fatty acid, and here the chain, that long chain, is the R, here is just represented by R's, that long hydrogen chain. And we can stitch it, we can do dehydration synthesis to one of these OH spots. We can make a covalent bond there. And so then we will have this glycerol with a fatty acid chain attached to it on one end. And this is called a monoglyceride, when it's got one fatty acid chain. And because we have these 3 spots, we can attach another fatty acid chain. There we go. Here's another one attached there, and we have a diglyceride. And finally, we can attach a 3rd fatty acid chain and make a triglyceride. And that's the maximum that we can do. We cannot attach anymore because there was only 3 spots to attach it. That's why fatty acids can't form polymers, because of this. And so this is a lipid. Now with 3 fatty acid chains, it's a triglyceride. Where are these used? Well, we can deposit this, we can use this, we put this into something called adipose tissue. This is the fat around your body. We can use it for energy. We can actually break this down and convert it and do all kinds of stuff to it and use it for energy, these glycerides. That's what we do with these fats. They also act as cushioning all over our body, especially these. And now these neutral fats, where is it? What can we talk about? Where's the thing about? Are called neutral fats. They're called neutral fats because where we had the fatty acid, so it was acidic, it released the hydrogen, but now we've taken that little bit away, and now it's got a pH of 7, so it's neutral. Monoglycerides, triglycerides, diglycerides are going to be pH of 7. And these will be stored as an alternative to glucose. Anything else I want to say here? Okay, so there's a variation of triglycerides, and it's very important. We're going to be running across this variation a lot. So what happens is, remember we've got this glycerol, which, there we go, let's just zoom in there. So we got this glycerol, we got those, 3 carbons with hydrogens, and then these OH, these hydroxyl groups, these OH groups. And so we can add 3 fatty acid chains. But what if we took one of these fatty acid chains? We got rid of the fatty acid chain. These are all also, remember, hydrophobic, and all of these things, they're all triglycerides, all of these things are still hydrophobic because those big long chains are hydrophobic. And then what if we added, instead of a fatty acid, on that third position we added a phosphate group and what's called what am I thinking of here? An amine, yeah, there's an amine here because there's a nitrogen, but this is a choline group. There we go. No, and basically, what you will notice here in this image, if you look in your notes, is there's the phosphate group. Look, the phosphate group has a polar moment. It's a little bit negatively charged. And this amino group is a little bit positively charged, which means that this region is polar. Remember, the rest of these, the tails, the fatty acids, are non-polar, therefore they're hydrophobic. But this polar head, now this polar region, this phosphate group, is polar, and it can mix with water. And this is very important, and you will find this molecule, which is called a phospholipid, all over your body, because it is the foundation of cellular membranes, all cellular membranes. Because what you can do now is, so let's say this is the polar phosphate head, these are the fatty acid chains that are non-polar, you can have another one here, and now you can make a whole wall of these, and the polar heads can mix with water. The inside of cells is water, the outside is water, but the inner region here is non-polar. Water can't cross through, dissolved things can't cross through, so you've made a wall now, and you've made a cell. And phospholipids will arrange themselves. They actually don't need a lot of help to do this. They will arrange themselves into this configuration with the tails facing in and the head facing out, the bases of all cell membranes. And then you can use proteins and have different mechanisms to move things across. You can control metabolism, you can control things going in and out of your cells and in and out of your body this way. So lipids, very important. Last thing, well, we can make waxes also out of lipids. And so when the glycerol, so when the fatty acids bond to something larger than a glycerol, it can make a wax, like earwax. We make those. And last is steroids. So steroids, if you lift, you probably have heard about steroids, taking steroids and things like that. And basically what they do is they are also non-polar, but they have a ring structure. Instead of being a chain, they are ring structured, and they can actually cross cell membranes very easily because they're non-polar. They can cross cell membranes, they have very powerful effects. So some steroids are hormones, and they have very powerful effects on the body. All kinds of things. Testosterone, I think, is a testosterone's a steroid. It was a hormone, and basically they activate parts of your DNA, so your body will start producing things that it normally doesn't. so muscle tissue, that kind of thing. But then there are always side effects to this, because it's not only the cells that you want targeted are going to be targeted. And one of these unimportant steroid is cholesterol. You may have heard of cholesterol. And basically, cholesterol is an integral part of cell membranes. It exists inside cell membranes, and what it does is it hardens cell membranes. So if you have a lot of cholesterol in your cell membranes, they are not as fluid, is basically what happens, and we'll talk about that when we talk about cell membranes. So proteins, to sum up, we've got the basic sort of building block of proteins is these fatty acid chains. They can come as saturated or unsaturated. They bind to a glycerol. You can have 3 fatty acid chains binding up to 3 fatty acid chains to a glycerol. So you have a monoglyceride, a triglyceride, a monoglyceride, a diglyceride, a triglyceride. These are used in fat tissue. If you replace one of the fatty acid chains with a phosphate group and a choline, you get a polar head, a polar region, and therefore you can make phospholipids, which are the basis of all cell membranes. If you have ring-structured lipids, those are usually hormones. Cholesterol is used in cell membranes to firm them up. They're also used as hormones to activate parts of DNA, essentially, to amplify parts of DNA. They're very powerful. Not all hormones are steroids. And I think that's it for lipids. So just, non-polar, remember those things. That's the big deals about lipids.